And Ken's going to talk to us about advanced affinity chromatography platforms for gene therapy and vaccine responses.
Thanks, Julie.
You're very welcome, Ken.
Oh, yeah. There you go. Good morning. It's good to see you all. I'm really excited to be here to talk about affinity chromatography and the affinity resins that we discover here at Repligen. So we're all here, more or less, because biologics don't purify themselves, which is fine. We have solutions for that. Typically, the bioreactors we produce these molecules in are very complex mixtures. And that means in order to go from the bioreactor to the drug, we have to pass through many steps, right? Existing state of the art, maybe you have a capture, a size exclusion, an ion exchange, a few things to get rid of all the stuff you're not looking for. And then at the end, you end up with your final product. But multiple steps means high drug loss or low drug yield. Each time you pass your drug through a column, not 100% comes out. If you can solve that problem, please come talk to me afterwards. Low drug yields means high manufacturing costs. You have to make more production to get the same amount of drug, pretty much no matter what it is. And the last thing we want is to have a drug program fail because of the purification process. It's just not something that needs to happen these days. That could be for two reasons. One, it could be a quality problem. So you have a product-related impurity or something that you just can't purify away from the final product. So I'm going to talk about how to solve that problem. There's also just high costs, right? If you have an extremely laborious purification for your very interesting biologic, it may not be commercially viable. We also want to avoid that. So Repligen is laser focused on this issue and basically lowering cost. That's the pointer, I'm sorry. Lowering cost by doing single-step capture with extremely high yield and purity. And so we accomplish that with affinity chromatography. And the goal here is to basically reduce your purification costs by tenfold by having a custom solution to the drug substance you're trying to purify. By doing just one step, you also save on development time, and ultimately, it's kind of the dream. It's like you have one chromatography step, you get 90% yield, you do a polishing step, and you're done, right? That's the goal. That's what we're trying to accomplish with our AVIPure resins, and I'm going to tell you about that today. First, let me just give you an example of how this could work for a challenging purification. So we had a partner come to us. They were purifying an FC fusion protein. They're capturing protein A, naturally. Protein A is great. Repligen makes a lot of protein A, and I don't have anything against protein A, but it has limitations. So in this case, the FC domains are kind of in this blue, and then the active portion of their molecules in green, right? And so the problem with protein A capture of an FC fusion is you get lots of product-related impurities. Anything that has an FC fusion is going to get brought along for the ride in that first capture step. And that's fine, but this ultimately led to them needing to reimagine their process. They can't just do this one-step thing we're looking for, right? Okay, so they go back, they do sizing, they do ion exchange, they add a bunch of steps, and they get to their drug substance. And this is the process that they had when they came to us, and the final yield was 25%. So what came out of their bioreactor, if for every 1 gram that came out, they got 250 milligrams purified. Unfortunately, that wasn't good enough for this product to be commercially viable. So they came to us and they said, hey, we know you can make affinity ligands. Can you purify this with yield greater than 25%? I was like, well, it's easy to step over a low bar, so let's do it. So we made an affinity ligand that purified their drug substance. It did so with high purity and yield of 90%, 93% in one step, okay? So we like to make sure that our partners are going to be able to do capture, polish, done. We also increased the capacity. So now they actually don't even need to use as much resin, or they can do more production for the same run. And we even increased the purity, right? Okay, so this is the kind of power of affinity, where the reason this works is because we aren't capturing all of those non-specific product target-related impurities. We're capturing just the final version that they're looking for. And during our affinity process, we hone in on exactly the drug substance that you're trying to purify so we can accomplish this goal. And it translates directly to cost savings, right? I'll mention this throughout, but ultimately, if you get 93% yield, That's going to save you money if your original process was 25%, right? So our partner was able to do 4 times less production to get the exact same amount of drug, and that was 3 to 5 times lower dollar per kilogram drug, okay? Everybody's dream. I've made a cool ligand, you've made a cheaper process, and we can go to the clinic. So how do we do this? Or maybe I should say, before we get to that, why don't we always do this, right? Why isn't it the case that if you have a drug substance, we develop a ligand for it, and you use that to purify it with 90% yield in 1 step? Why not? Well, it takes time, right? It's not like your process is going to develop itself overnight. You already have a 25% 5-step process. It works. If you just make more, you get what you need, right? So that's one thing. I'm going to talk a lot about how fast we are, so hopefully I can allay that concern. There's also, of course, the risk it might not work, right? Maybe you have made a biologic that is truly impossible to develop an affinity ligand for. If so, you'd be the first, and I'd be happy to come talk to you since I'm always looking for a challenge. Ultimately, we haven't yet found a biologic that we can't purify with an affinity ligand. So let me just off the bat say it's not a risk. We'll find you a ligand. Of course, there's cost. As much as Repligen would prefer, I don't work for free. So I think if cost is a concern, let me also allay those. We make sure we always price competitively. And you can talk to many of my colleagues here. We want to make sure that we can provide a solution that's going to work for you. And then lastly, supply chain, right? Protein A is something that Repligen makes a lot of. Repligen's really good at making affinity ligands. So if you're concerned about your supply of affinity ligand, again, we can solve that problem for you. A lot of what you're going to hear about today is kind of the robustness of the vertical platform that Repligen has built in order to supply things like affinity resins, okay? So time, risk, cost, supply, forget about it, and listen. Listen to the story of affinity resin chromatography, and think about, as we go, what you're working on. What's your biologic? And could this solve that problem? Okay, so let's start with time, and also how we do it, since this is where I get really excited. I know Julie I only had 1 cup of coffee, so that's the good news. I'm not going to run off the stage. So we can do this whole process, take your drug substance, find an affinity ligand, and give you a prototype resin. We can do that in 3 months. So if you came to me in the back, and you snuck me a vial of your hard-to-purify drug substance, I could have you an affinity resin prototype in your lab at R&D scale at the end of October. Now, your legal department might not like that, so maybe we should let them do some paperwork first. But actually, no joke, sometimes the legal paperwork takes longer than the affinity resin discovery. So let you ponder that. Why is that the case? That's because we've built a huge platform of ligands, which I'm going to talk about in a second. And we narrow it down from about 1 billion candidates to about 100 in just 3 weeks. Then we take those 100 and we actually do process development on a whole swath of potential candidates. And we dial in to the client's needs what the final ligand will be. So all of these 100 candidates capture the ligand, or sorry, all of these 100 ligands capture the drug substance. That's not the problem. The problem is, can we develop a process that with one of these affinity ligands will meet the commercial needs of the partner? So if you say, okay, I have all these product-related impurities because I have an Fc fusion, then we make sure we find a ligand that doesn't capture those. Oh, I have lots of HCP, or I have lots of DNA left in my purification. We make sure that we dial in that process. And that's because we're screening even on resin, a wide range of candidates. So then we take the methods that we've optimized. All that data, you get that data. So maybe you don't believe me when I say, look, this is the best process for this ligand, this affinity resin. Well, that's fine. You have all of our data. You can take it and you can work on it in your lab. And you're going to get that in October if you give me your drug substance today, right? So you both get the resin and you get the method. 3 months. Then you decide, wow, Ken has made a really great resin, and I'd like to scale this up. That process of going from an R&D scale resin to a cGMP ready for your clinical batch resin is a little less than 6 months. And so that process ultimately is something our partners often opt into at risk immediately because their timelines are very quick, and we're happy to do that. So as soon as we're done generating a resin, we can start the transition to commercial supply. And what that means is that in less than 3 quarters, you could have a resin that is ready to go in your process. And, of course, because Repligen has a wide range of column sizes and offerings with the Opus platform, you can get it at kind of in any format you want, with all of the compatibility and the reliability and the pre-qualification of the Opus platform. All of that comes with these affinity resins, except it doesn't have to be protein A now. It can be something that's custom to your drug substance. So this is ultimately an unrivaled timeline. As a discovery scientist who has worked at multiple different companies, I can say we have taken every shortcut there is to take in ligand discovery to find you a ligand in the fastest possible time. I'm going to talk a little bit about how that works in a second, but I just want to also reiterate that we have not yet failed to find something, okay? And these two things are not unrelated. So it's not that we're really fast and we never fail. We're really fast because we never fail, and part of that is because of the ligand library that we've built. Oops, sorry. So the key to this whole thing, the linchpin of the entire platform, is the proprietary ligand scaffolds that Repligen has identified. So if you think about a cell, right? It's got tens of thousands of proteins. They're all mixing around, but they're all accomplishing very specific biological purposes, right? So some are binding to membranes, some are binding to proteins, some are binding to DNA. What we've done is we've gone through and taken protein folds and peptide sequences out of nature. We've engineered them to optimize for bioprocess, so things like sodium hydroxide stability, the ability to refold on a resin, things like that. And we've built ligand libraries out of them. And so we screen, every time we take in a drug substance and try to identify a ligand, we screen 48 different scaffolds. That's compared to our competitors, who I will just say are perfectly earnest in their attempts to find an affinity ligand with their 1 or 2 scaffolds. There's nothing wrong with that. But if those 2 fail, what are you going to do? I guess you're done, right? Where we have 48, and actually I've seen this happen. If 47 of those fail, that's fine. We have 1, and we can take that 1 and we can iterate on it. We can mature the affinity. We can change the sequence. We can identify something that will get you to an affinity resin, right? So the ability to have both a wide range of ligand scaffolds and also to just leave the 47 that don't work and move forward with the 1 is what allows us to get this kind of paired speed and success rate, right? And I want to emphasize, this is not just like a law of large numbers. We're not just like screening everything that exists. We've identified these scaffolds to very specifically address every biologic that we know is going to the market or is on the market. And it's the beauty of the natural protein, the proteome, is that it's essentially solved how to bind and let go of all the biological molecules we might want to use as drugs. So all we're doing is we're taking a wide range of protein folds and we're engineering them for bioprocess. To be clear, this took like 10 years. So when I say all we're doing is like 10 years of my colleagues' work. It's not something you can build overnight, and we're really proud of it. What it's done is it has allowed us to essentially purify anything. And again, I'll reiterate my challenge to you. If you have a biologic you can't purify, I'd like a crack at it, because I bet we have a ligand in our libraries that could purify it. And purely from an intellectual curiosity point of view, I think we have what it takes. We have done this in the past, so you don't have to take my word for it. We've done 30 different development programs with over 20 partners now. I'm going to focus today on vaccines and viral vectors. Those are the kind of most interesting new things that we have in our pipeline. I'm also going to touch on all of our products at the end. And if you're curious, we have a poster and some bottles and resins and things. I'm also just happy to talk shop in the back afterwards. If your drug isn't on this list, that's okay. You still belong. We've purified everything, really, from hormones, enzymes, recombinant proteins. We even take bispecific antibodies and purified just the bispecific, not all of the other stuff you're not looking for. Again, with a 100% success rate. Okay, so with this platform, what we've done is basically offered three things to our partners. One is kind of what I've been talking about. It's this custom thing. So you have a drug substance, you want an affinity resin for it. And so you come to us, you take your drug substance, 3 months later you have a prototype resin, you're testing it in your lab, and after we transition it to cGMP, you get it in your OPUS columns, and so one resin, one drug, okay? The second thing is actually something we've seen more and more from our partners, and it's what I like to call a platform resin. So it's where you're developing a wide range of biologic, but they all have something in common. So, you know, they're all, to use the easy example I keep referring to, they're all FC fusions, right? But you're changing kind of the portion of the molecule that's actually causing your therapeutic effect. And so in this example, you know, you've got your green and your purple drug, and you've changed a little bit down here too, but we've targeted an affinity resin to this top blue domain here. So you can purify anything that has that domain, right? And it's still all the same rules of the road. So you have to give us those drug substances. We'll find an affinity resin to the specific domain you're looking for. 3 months until you get your prototype. OPUS columns after you have your cGMP transition 6 months later. It's all fun and games. Now you can purify multiple drug substances with your same affinity resin. So if that's something you're working on, we can totally do that too. And then last, and probably the most interesting and exciting, is what we're calling our catalog resins. So this is not proprietary. So this, you don't have to come to me and keep emailing back and forth and actually have us do work. We've already done the work, right? So we've gone out into the field and we've identified what kind of the hot, trending biologics are, and we're building affinity resins for them in advance. And that means they're available today. And they're available to everyone, not just the person who came to us first and tried to get a license for that resin, right? Generally, we design these to be specific for a modality. So I'm going to talk about vaccines and AAVs today as an example. It also means you get your cGMP resin today, because we've already transitioned that internally before it ever goes public. You know, so you can get your OPUS column today. You want a sample of AAV2 or AAV8 or AAV9? They're in the back. Well, I can't give you the ones in the back because I have to show them to people, but we'll ship you one, okay? This is the thing I'm most excited about, because honestly, there's a lot of really interesting classes of biologics that could really benefit from this. And so we're taking those in, we're developing affinity resins, and we're launching them as fast as we possibly can. So I have to say, every time they have me come talk, it's 1 day slower. So I don't know, maybe. What is Repligen's real goal here? All right, let me first talk about vaccines. I really like this project. It's near and dear to my heart because it's something that, of course, we've all experienced over the last few years. And also because it's an interesting challenge. Viruses change over time, right? Ultimately, if you've developed your downstream process for the original variant of COVID, it's possible that that doesn't work anymore on the ones that are circulating now. And that's a unique challenge. Oftentimes, you get to decide what your drug's going to be, and then it stays the same through the whole process. But ultimately, if that same molecule is out in the world changing, you're going to need a platform approach. And so we thought about this very carefully, because it's not obvious if you can't sit COVID down and be like, stop changing here so we can develop an affinity resin here. You can change over there, fine, no problem, right? And what we ultimately settled on was using a scaffolding domain. So that when you're developing a recombinant protein vaccine, as long as you incorporate a scaffold that is meant to drive trimerization, we could purify off of that. So if you're familiar with the field, T4 fold-on is probably the most common trimerization domain that is used. It's fascinating. It's encoded in the mRNA vaccine. So you have, in an mRNA vaccine, you actually have the spike protein, a linker, and then T4 fold-on. And what T4 fold-on does is, even as it's produced in your own body, it forces the trimerization of the molecules so that it's presented correctly, so you'll raise the correct immune response, right? So it works literally in us. It also works in a bioreactor. And so that's why people take T4 fold-on and they fuse it to their vaccines. And so many, many, many recombinant vaccines use T4 fold-on trimerization domains. If you're one of the people who is working on one of those, I have good news. We have a catalog product. It's coming up next year. So I can't ship it to you today, but I'll talk to you about it today. So I have some data on my poster in the back about this resin, and I'm really excited about it. I think it'll help solve a lot of the mRNA versus recombinant protein speed problems, right? So if you don't have to develop a new downstream process, that is like instant time savings, right? The mRNA people, they just get to synthesize, send the polymerase on the way, and then, and then they're off to the clinic. With proteins, it's nice to have something comparable to that. That's what we're working towards. So T4 fold-on is awesome. A lot of companies also have been doing proprietary trimerization domains. So I just want to give you one example of kind of the power of affinity before I move on to AAVs. We had a partner come to us, and they said, OK, we are going to make vaccines for viruses that don't exist. I said, OK. Well, how's that going to work? I say, OK, well, we're going to make a proprietary trimerization domain. It's going to be only like 1% of the surface. I need to stand a little more in the middle. There's going to be like 1% of the surface of the antigen. And we want to make sure that your resin is going to purify any antigen that we attach to that. So we're going to give you 5 antigens. And we're not going to tell you what they are. So good luck. Also, by the way, they can't be eluted in the same way. Some have to have high pH, some have to have low pH, because of course, you don't know what the antigen is going to be if it doesn't exist yet, right? They're going to be a wide range of sizes, 60 to 600 kilodaltons. But you have to provide us with one resin, high capacity, high purity, one step, okay? So that was the challenge they gave us. And I'll admit, our timeline slipped 1 month. But in 4 months, we delivered this process to them. And so here I'm showing you a breakthrough curve. If you're not super familiar, it's pretty straightforward. We basically take a column, and we start loading in drug substance until it flows out the bottom. And that basically means we know exactly the maximum amount of drug substance that can bind to the column. And then when it starts flowing out the bottom, the curve goes up. And we call that the capacity. So this antigen 1 had a capacity of around 5 grams per liter, maybe like 5.2, since we say 10%, DBC 10, right? Antigens 2, 3, 4, and 5 were a little bit higher capacity. And I was like, put on my chromatography thinking cap, and I was like, I bet antigen 1 is higher molecular weight. And sure enough, yeah, so this is the 600 kilodalton construct, which means just less molecules will fit into a packed resin bed, right? That's kind of the basic rules of chromatography. And these are the smaller ones. Ultimately, their goal for this antigen was 3 grams per liter capacity, and we provided 5. So even for these really large molecules, we're always meeting specification for capacity. I was really proud of this project. Not only did they give us not the sequence, and they gave us only the trimerization domain, but they covered the trimerization domain in sugars. And then they said we need a specific glycoform of these different antigens. So we were able to accomplish all of those goals. And they're off to the clinic, which is something I'm really proud of. OK, so I'm going to transition now to gene therapy. I think it's something of high interest to everyone in the room. And AAVs, because we have AAV products, is where I'm going to focus. Before I get into AAVs, I'll just mention we have a lot more coming. So if you're not working on AAVs, but you are working on gene therapy, again, come find me in the back. I'm happy to tell you at least a little bit about what we're working on. Spill a few secrets. AAVs are really interesting, though, because it's the kind of thing where we weren't totally sure they'd be great drugs until super recently. There was early clinical data that kind of cast doubt on the modality as a whole. And that meant that a lot of the trials were relatively low patient population, and the upstream processes weren't optimized. There was no need. There just wasn't a huge demand for particles. And as the field has matured and the really promising data has come in, ultimately that's led to larger patient populations. I mean, there are hundreds of AAV trials running right now. That's led to intensified upstream processes. Someone was just telling me they're going to do a 5,000 liter AAV production. You're going to need a good resin to purify a 5,000 liter AAV production, right? Ultimately, people are also engineering their capsids as they try to develop specific targeting mechanisms or whatever kind of specific gene therapy they're trying to accomplish. This has meant that ultimately the bioprocess of AAVs hasn't quite kept up. So we really need to mature in the purification processes of AAVs in order to keep up with the experiments that are running in the clinic. And again, as I said at the beginning, we never want purification to be the reason that a trial doesn't run or a drug can't be sold. And so in order to fill that gap, Repligen has developed the AAV 2, 8, and 9 serotype-specific affinity resins. A couple of really cool things about these resins. One is they're cleanable in NaOH. So it's the first AAV resins that can be cleaned with sodium hydroxide, kind of the gold standard clean in place. They're also serotype specific. If you're interested in serotypes other than this, I've got good news. Those are coming too. So come find me afterwards. I want to show you a little bit of data from a couple of these resins, since I really think they're designed to meet the bioprocess needs that we see in the field today. So traditionally, in AAV, you have a relatively low virus titer in your production, and that leads to either the need to make a lot of production or to concentrate that production or some kind of combination of those two things. So we wanted to solve two problems when we developed these AAV resins. One, we want to completely avoid pre-concentration. You should never have to concentrate your lysate. It's just not necessary, right? We can develop a resin that can capture very low titer productions. Also, you just shouldn't have to run for 3, 4, 5 minutes. We should be able to develop a resin that's high enough capacity to avoid residence times of more than a minute. And so that's what we made these resins with. That's what we had in mind when we made these resins. I'm showing you here that same breakthrough curve, but now instead of 5 different antigens, you've got 4 different bioprocess conditions. And so the one in the middle, this orange one, is something that I would basically call a realistic AAV purification, where you have only 5 to the 12 virus particles, and you are only doing a residence time of just 1 minute, right? And even at that kind of realistic curve, a realistic condition, our capacity is around 5e12 virus particles per milliliter of resin. So this, I think, is more than enough capacity for any AAV production I'm aware of. So if this wouldn't meet your needs, I'm actually super interested in hearing about that. I think capacity is something that all of our AAV resins are kind of far exceeding what the productions are like. So that's one thing. And then, of course, I want to reemphasize this cleaning in place because our direct competitors in the AAV field do not offer this, right? So you cannot clean other resins with NaOH, but we have built in to our ligands the ability to withstand high concentrations of hydroxide. So this is the advantage of building your library from scratch, right? So when we take a certain affinity ligand out of nature and we optimize it for bioprocess, what we do is we remove liabilities. So like asparagine's gone. Don't need those. Hydroxide can basically completely clip them off, right, and ruin your function. That allows us to build these resins that are super stable to sodium hydroxide. So this is an experiment where we've done an AAV8 purification with 20 clean-in-place cycles at 0.5 molar sodium hydroxide. And the chromatogram here is pretty compelling. The bottom one is the naive resin, never been cleaned. And then this is kind of the binding and elution and wash, wash, elution, and strip. And this is 5, 10, 15, and 20 cleaning in places with 0.5 molar sodium hydroxide. And you can see there's essentially no change. So no change in yield, no change in purity, no change in DNA concentration. It's the same. Okay, and so it's built into the ligand. It's built into all of AviPure resins. We're talking about AAV now, but if we do a custom project for you, same thing. We'll make sure that this is a feature. And of course, we're familiar with the benefits of CIP with sodium hydroxide. But one thing you may not think about is just as you scale up to larger and larger batches, the ability to reuse resin. Of course, at scale, 10 reuses is way better than 3, and 20 is way better than 10. Also, you don't have to open your system, right? So if you're doing a large-scale purification, you can take the same large column and run 20 purifications over it before you have to open anything or change anything. Just set it and forget it. And that's a huge advantage at really large scale when there's kind of a lot on the line. Of course, it simplifies R&D. This is the world I like to live in. So we can test our resins many times on many different conditions, and we can clean in place and be confident that we're doing the same experiment. We're not doing some kind of, look, as we did these 20 experiments, we saw drift, and so our toxicity is worse than the last lot and then the first lot, right? We can really avoid misleading data early in the process by having a really clean resin. And of course, bioburden. Nobody likes bioburden. cleaning in place is ultimately the answer, and that's why we build it in. Okay, so Russ just told me I only have 5 minutes left, and I really wanted to pontificate on my last slide here. So I've told you about AAV2, 8, and 9. They're really awesome resins, and they purify AAV2, 8, and 9. If you're not working on those specific serotypes, I've got good news, which is that we're working on a whole range of other gene and cell therapy modalities. So we have a CD81 specific exosome resin that's in the pipeline. And actually, one of my favorites is this phospholipid resin. So we're purifying on the membrane so that you can capture extracellular vesicles, kind of regardless of the proteins that are in them. Of course, we have other serotypes. We have AAV5 and AAV6. We have VSVG lentivirus. So that ligand is in production. And a bunch of other interesting things. I was talking to some mRNA people. So we're working on mRNA as well. Along with additional exosome markers. All of these custom affinity resins will be available in all of the Opus formats. So if you're thinking about, is it worth testing, I think the one advantage is that you don't really have to just wonder what it would be like to have an affinity resin a year from now. You can talk to us, and we'll get you a prototype in a robocolumn as soon as it's available so that you can start integrating into your process. And I recognize that a lot of times, you have a process that works. You know what, 25% yield, it's fine. It gets the job done. So we want to make sure we meet you where you are. We can do better than that, and if getting you something early in your process as a prototype resin will help accomplish that goal, we're very open to that. I'll also say, if I haven't mentioned the biologic you are working on, one more time, come find me. There's more things that we're working on than I'm allowed to list on the marketing slides, and so let's talk, okay? It's been a real pleasure talking about our affinity resins. I hope I've given you a good overview of what's possible with affinity chromatography, what's available today, and what's coming soon. And so with that, I'll hand it back to Julie to introduce our next speaker. Thank you.
Thank you. Before you go, 100%. That's,
yeah, actually,
did you hear that? 100% success rate.
I'm waiting for that number to drop. I genuinely am. I kind of want to see what we can't do. So come find me.